With this talk, we'd like to provide some updates on fungicide resistance management and weather data measurements from canopy level sensors and how that is related to disease infections compared to weather data from on-farm weather stations. Now, this talk is prepared by myself and Ms. Anita Schoeneberg, who currently works in our lab and is in charge of our ongoing field trials, and to better understand row cover effects on weather variables and disease infection risks. When it comes to strawberry diseases, anthracnose probably has been the major issue that could wipe out the entire field if control measure is not effective. As most of you are aware, there are two species complexes responsible for the disease, Clototrichum acutatum and Clototrichum glodosporides. Now, disease infection is usually favored by extended moisture and warm conditions. Typically, the disease pressure increases as season progresses, especially during fruit ripening and harvest. Symptoms normally refer to black spot on upper parts of the plant, blackened seeds, and bright orange sporulation can also occur on mature fruit, as shown on the picture to the right. It's also important to note that crumbs can also be affected, causing the crown rot. With regard to source of inoculum, nursery transplants have been considered the major source. In addition, some weeds may also harbor the pathogen that could sporulate and cause the infection under some conditions. What Clototrichum species do we have here? Now, as we just mentioned that strawberry anthracnose could be caused by either Clototrichum acutatum or Clototrichum glodosporides. Traditionally, acutatum was thought to be the cause of fruit rot, whereas glodosporides was the cause of crown rot. However, most recent studies have confirmed both species being the complexes, meaning that there are different species under each complex. Now, in the past several years, a total of 200 isolates of Clototrichum were collected from strawberry fields in the mid-Atlantic. While most of them were from Maryland, we also had some from Pennsylvania, Virginia, and North Carolina, which gave us an insight into the species diversity. Consistently, as you can see, Clototrichum nymphaea was the most prevalent species across the states. Looking at the species from different host tissues, for those from the fruit, 95% were Clototrichum nymphaea and 5% were Clototrichum fiorinae. With regard to isolates from the crumb, 49 was nymphaea and 49 was Clototrichum siamense, which belongs to glodosporides complex, and the Clototrichum lineola only made up 1%. As for the isolates from the stem and runners, all of them were found out to be nymphaea. Now, however, most of our isolates were from the fruit, and Clototrichum siamense, again, that's species under glodosporides, would be expected to be more prevalent if there are more isolates from the crown. Now, based on this, Clototrichum nymphaea seems to be the predominant species responsible for fruit rot as well as stem and runner infections. With regard to crown rot, both siamense and nymphaea are primary pathogens. Here is a list of conventional fungicides that are currently labeled for strawberry anthracnose control, with the information of active ingredient, trade name activity, whether they are systemic or contact fungicide, efficacy for anthracnose control, as well as resistance risk. For example, the first one, cyphenomethyl, that's the active ingredient in the product Topsin M, which belongs to group 1. As it's shown here, the most effective fungicide group for anthracnose control would be FRAC 11, strobilurins. However, now resistance risk is very high. Continue using this chemical group is not recommended. At this point, we believe that Switch and Miravox Prime would be the best materials for anthracnose management. Both products contain fluvial X-onil, now followed by Captan and certain DMI fungicides such as Tilt, Inspire Super, Quilt XL may also have some efficacy towards anthracnose management. However, except for Captan, all other fungicides are at risk for resistance development. Under low disease pressure, Captan is recommended because resistance development is unlikely to occur. Early in the season, Thiron could also be useful due to its higher efficacy for botrytis and can also help with deer management. Please note that cyphenomethyl is ineffective against acetatum, whereas fluodeoxonil, however, may be weak for Calototrichum glodosporides. As we just mentioned that the risk of resistance to FRAC 11 strobilurins is high, we also detected resistance in those 200 isolates from the region. As you can see here, the resistance is widespread. Each circle represents number of isolates from each location, and that the proportion of isolates with resistance is indicated by black or gray color. Overall, 40% of the isolates were resistant to strobilurins. Now, most of our isolates were collected between 2014 to 2020. We did find an increased frequency of resistance in isolates collected more recently. Last year, we had a fungicide efficacy trial to evaluate certain fungicides for efficacy control, including Captan, Mirovus Prime, and Merivon. Now, both Captan and Mirovus Prime were found to be very effective for anthracnose, with less than 5% disease incidence occurred in those plots. However, Merivon, which was frequently used for anthracnose management, had almost low efficacy compared to the control. Presumably due to the resistance issues, Captan worked unexpectedly well in our trial. In part, could due to the fact we had a weekly spray program where Captan was used preventatively. Now, if the disease has already occurred in the field before Captan was applied, the efficacy could be much less because Captan is a contact fungicide that has no kickback, no systemic activity. To conclude, resistance was found to be widespread to strobilurins, FRAC 11 fungicide group, which may no longer be effective for us. Fluodeoxonil, however, is very effective. Now, this active ingredient is available in both Switch and Mirovus Prime, but it's important to note that fluodeoxonil may be weak against Calototrichum glodosporides complex, causing the crown rot. Both Captan and Thiron, they're much less prone to resistance development. Also, they have some efficacy for Calototrichum species, which should be utilized more frequently for resistance management, especially under low disease pressure. Propiconazole, difenoconazole, those are FRAC 3 fungicides. They have shown good control efficacy based on in vitro and detached fruit assays. Interestingly, other active ingredients within the same chemical group are not as effective as those two fungicides. The figure below showed EC50 values of different Calototrichum species causing strawberry anthracnose for different active ingredients within DMI fungicides. As you can see, propiconazole and difenoconazole work very well across all those species, whereas other DMI fungicides are weak for certain species. Gray mold is another important fungal disease on strawberries, which typically drive spray programs during the season. Now, the disease is caused by two different species within Botrytis genus, Botrytis cinerea and Botrytis figariae. Interestingly, Botrytis cinerea was able to cause infection on both the flowers and the fruit. Botrytis figariae, though, it's pretty limited to the flower infection. Now, both species, they have great ability to develop resistance. Similar to anthracnose, extended moisture would favor the disease infection. Here are the conventional fungicides labeled for strawberry gray mold control. Similar to the resistance issue we have in anthracnose pathogen, high frequency of resistance has been detected in a number of fungicide groups labeled for botrytis management, such as Topsin M, Rovra, and Elevate. Therefore, continuation of using those fungicides is not advised. In general, newer Group 7 fungicides such as Lula Tranquility, Sensation, Marivan, or Group 7 fungicides including Switch and the Mirovos Prime would be the best materials at this point. Again, due to the nature of single-site fungicides, they are unfortunately all at risk for resistance development. Both Thyran and Captan unlikely to develop resistance. They should be a backbone of your spray program. Now, in general, Captan has better efficacy towards anthracnose, whereas Thyran has a better efficacy towards botrytis. This figure probably has been showed a couple times already, but I just wanted to show it here again, just to emphasize the importance of resistance management. Now, this was a resistance monitoring program we had for botrytis, and we did find an increased level of resistance to different chemical classes, fungicides commonly used in strawberry fields over the years. I wanted to point out though, probably you can see here that there were two spikes in terms of frequency of resistance to fludial X-onil during the 14 to 15 and the 15 to 16 seasons. Now those spikes were mostly due to the increased frequency of resistance in Botrytis figariae. If you remember, we mentioned that Botrytis cinereae and figariae, those are two species that could cause strawberry gray mold, and we found that Botrytis figariae is more resistant to switch to fludial X-onil. Now this pathogen is a primary flower pathogen, pretty limited to flower infection. Now this could suggest that fludial X-onil switch or Mirovos Prime may be better to be used later in the season for botrytis fruit rot. Now we have talked about frequency of resistance, percentage of resistant populations, but what do they really mean? 10%, 20% resistant, right? Is there a resistant frequency threshold at which fungicide efficacy is compromised? There is no solid data. Based on our preliminary experiment, we think that over 50% efficacy reduction may occur when just 20% of isolates in the field are resistant. Now this means that mixing with protectant fungicides such as Thyran or Captan may provide uncontrolled security when resistance situation is unknown. However, when resistance has emerged, it can be selected rapidly by the respective fungicide regardless of mixing with multi-site fungicides. Now, if you know your resistant situation, then you don't have to time to mix with Captan or Thyran each time. With more frequent rainfall events are predicted in the East Coast, I thought it may be helpful to offer some tips for fungicide applications. Protectant fungicides such as Captan and Thyran, they are generally good for about 6 to 10 days. Systemic fungicides good for 8 to 12 days after application. Of course, depending on weather conditions. For protectants, it should be reapplied after 1 inch of rain if the residue is 5 or more days old. For systemic fungicides, consider reapplication after 1 inch of rain if the field has not been treated for the past 7 or more days. However, if dry weather is forecasted, the spray intervals could be extended since the dry weather is not favorable for pathogen infections. Most systemic fungicides are rainfast after a few hours, but may take up to a half day for others, including protectant fungicides, to dry. Importantly, we wanted to spray prior to rain events if possible. Why? Because under wet conditions, infection risks would increase significantly. Both protectant and systemic fungicides are effective when applied before infection occurs, but only systemic fungicides have some kickback activity. So if you apply protectant fungicides after the rain, there will be pretty much low efficacy. Apply fungicides after the rain will also deal with increased level of inoculant because you allow the pathogen to establish, that's reducing fungicide efficacy and increasing selection pressure for fungicide resistance as well.
My name is Anita Schoeneberg, and I'm a technician in Dr. Hu's lab. Today, I will share with you some exciting results from our ongoing trials with microclimate sensors at the strawberry canopy level and their use to improve disease prediction precision. Botrytis and anthracnose fruit rot are important diseases in strawberry production. The infection risk for those two diseases can be predicted using disease models that are based on leaf wetness duration and temperature. The so-called Strawberry Advisory System has been developed in Florida and was really successfully implemented. Fungicide use could be reduced by 40% on average compared with weekly calendar sprays, while no significant differences in marketable yield were observed. In our trials in the Mid-Atlantic between 2017 and 2019, we also saw a reduction in fungicide sprays by 30%, while marketable yield and disease incidence were largely comparable with the weekly calendar spray. So we saw that the Strawberry Advisory System can generally be implemented in the Mid-Atlantic. However, the growing conditions in the Mid-Atlantic are quite different from those in Florida. The most obvious difference is the use of floating row covers. Those row covers can be used in winter for frost and freeze protection, but also in fall already for flower initiation and crown development promotion. Obviously, an on-farm weather station cannot account for the microclimatic conditions under the row covers. And even without the covers, the microclimatic conditions can be quite different in the canopy. Just think of like shading between the plants. On the right-hand side, you can see a weather station that is traditionally used to get the weather data. In this case, it's right next to our experimental plot, but that's not always the case. A weather station can be miles away from a farmer's field. So we combined this weather station with microclimatic sensors that you can see in the picture below. This microclimatic sensor set is composed of two components, the leaf wetness sensor that you see in the red circle, and the sensor that records parameters like the air temperature or the relative humidity. The leaf wetness sensor is again in the larger picture. It basically looks like a leaf and acts like a leaf. So whenever the sensor is wet, the leaf is wet as well. We think that by monitoring those canopy level environmental variables, we will significantly improve the disease prediction precision. This is what the experimental setup looks like at one of our sites. On the tripod you see the weather station, and then labeled with Canopy 1 and Canopy 2, those are the microclimate sensors at canopy level. We are conducting this trial at 3 experimental sites over 2 seasons, at the Y Research Station in Queenstown, Maryland, and then we have 2 commercial orchards, one in Maryland and one in Virginia. The treatments for the timed fungicide sprays are based on 4 timings. The first one is a weekly calendar spray roughly on the same day every week. And then we have 2 model-based treatments. The first one gets the data from the weather station, which will be called Ms on the following slides, and the other treatment gets the weather data from the canopy level sensors. And of course we have an untreated control. For the harvest time, we will do weekly evaluations of anthracnose and botrytis incidence, and we will also measure the marketable fruit yield. For the data visualization in real time, we are partnering with the platform called XZoom. You see here an output of X-zoom in the first picture for the disease infection risk. As soon as that black line enters the yellow shaded area in the graph, this is a medium infection risk, and this would call for a spray that has to be applied within 48 hours. Obviously, red means a high infection risk, so we can actually see how high the risk was for this infection event looking back at the spray timing. X-zoom can also be used to show other parameters that can be calculated from the weather data, for example, the degree days that you see in the second chart. So this offers a lot of benefits for the grower. This is the temperature output from X-zoom for a period in spring in one of our Maryland experimental sites last year. The time frame is from April to May, and maybe remember those cold snaps that we had during this time last year. Between the blue arrows you see periods where the row covers were on, so the covers were taken on and off several times depending on the freeze events that were predicted for the night. In red you see the temperature that was measured by the weather station, and in orange and yellow the temperatures measured by the two canopy level sensors. You can see that during night, mostly the row covers were able to increase the temperature in the canopy, which is what we expected. But what I want to show you here is the striking differences in the temperature during the day if the cover was on. So especially in those two time frames at the beginning of May, you see that the temperature underneath the row covers reached almost 100 degrees Fahrenheit during the day. And during the night it was below 40. So those are like just huge differences that the row covers can make. And this also shows why it's important to remove the row covers during the day if a sunny day like later in the year is predicted, where the sun has a lot of strength and makes it very, very hot underneath those covers. The second thing I want to point out here, which you can see in the green circles, is that the temperature in the canopy is not always higher than at the weather station. So it seems like, especially during the night when the rows were not covered, we recorded even lower temperatures close to the ground where the canopy level sensors are compared with the weather station. This figure shows the average temperature under the fall cover at the Y. At the Y we have a special experimental setup. We have two identical plots, and one of those was covered during fall while the other one was uncovered. In purple you see the air temperature measured by the weather station, and then for the blue and the green lines you have to refer to the y-axis, which gives the difference in temperature. So whenever those lines are above 0, it means that it was actually warmer. The temperature recorded by those sensors was higher compared with the temperature measured by the weather station. Between the two red arrows is the period where only plot 1, which is represented by the green lines, was covered. And it's obvious that the temperature was higher than in the uncovered plot. After mid-December, both plots were covered, and mostly the average temperature was higher under the cover compared with the weather station. But there are also times, as we also saw in the previous slides, when the average temperature during the cover was actually lower than measured by the weather station. This chart is again about temperature, but this time we are talking about the maximum temperature. So the maximum temperature is the highest temperature that was recorded for a day within a 24-hour time frame. This is for one of the sites in Maryland, and again between the two blue arrows that's the period that was covered. And in this case, purple is the weather station, and orange and red are the two canopy level sensors that we had installed at this site. And then the bars that you see at the bottom, the blue and green bars, they are the temperature difference. So whenever this bar is pointing upwards, the temperature in the canopy was higher compared with the weather station, and when it's pointing downwards, it was lower. And you can see that the maximum temperature is always higher in the canopy, independent of the use of the row cover. But during the covered period, you can see that this difference is more pronounced, at least in the winter months. When the cover was removed in March, there's still like a slight difference, but in general the temperature in the canopy increased in March when the temperature in general was rising. This is the same site in Maryland, but this time the minimum temperature is displayed. The minimum temperature within a 24-hour time frame is often the temperature early in the morning just before the sunrise. And we see that the minimum temperature before the covers came on was often actually lower in the canopy compared with the weather station, and the same pattern after the covers came off again. During the covered period, it's sometimes higher and sometimes lower in the canopy under the covers, and this seems to be connected to the general temperature. So whenever the temperature was rather low, the covers had an insulating effect and the temperature underneath the covers was higher compared with the weather station. But when the temperatures were a little bit higher, the temperature at the weather station was actually higher than the temperature recorded at the canopy level sensors. So for the minimum temperature, it's not always higher in the canopy or underneath the covers. In the beginning, I said that the model works with two input variables, which are temperature and leaf wetness duration. We saw that the temperature is increased in the canopy as well as under the row covers in comparison with the weather station. And now we want to take a look at the leaf wetness duration. So in this chart you see again in purple the leaf wetness duration measured by the weather station, and then in red and orange the leaf wetness duration measured by the two canopy level sensors. The red sensor is in the inner row and the orange sensor is in the outer row of the field. I subdivided the whole time frame into several times where we can see some trends. So the first two time frames are from October to November and from March to June. Those are the two time frames when the plots were not covered. And we can see that the leaf wetness duration was higher in the canopy compared with the weather station. The covered time frame was from November to mid-March, and I subdivided this time frame into two separate smaller time frames because of the weeds that we saw when we uncovered the plots in March. So we thought that the weeds might have had an effect on the leaf wetness duration underneath the covers, because it looks like, especially from January to March, that leaf wetness duration was greatly increased by the covers. But we think that this is mainly because of the weeds, because when we look at the time frame from November to January, when the weeds were probably still small and dormant, we see that the leaf wetness duration is still increased, but not as much as in spring when the weeds started growing. So we also have some preliminary data for this season, and we see the same trend. While the plots are uncovered as well as while the plots are covered, we see an increased leaf wetness duration in the canopy. So concluding, we can say that we don't really see an effect of the row cover, but the leaf wetness duration is higher in the canopy. Also, the inner row, which is more protected, has a higher leaf wetness duration than the outer row. As I said before, we also have a site in Virginia. So those are the same chart leaf wetness duration in Virginia for the two seasons, and we see that we have roughly the same picture. So independent of the row cover, we have an increased leaf wetness duration measured by the canopy level sensors. And this is true for both seasons, although the 2020-21 season is still preliminary. We don't see a large difference between the two sensors, but there is a certain variability as well. This is the leaf wetness duration at Y, our third site for those trials. The Y is special because we have two identical plots, as I mentioned before, one covered during fall and the other one not covered during fall. Both plots received the cover for frost protection during winter. So looking at this chart, the weather station is again purple, and then the two green bars represent the inner and edge rows for the covered plot, and the blue bars represent the inner and edge rows of the uncovered plot in fall. If we look at the uncovered period between October and November, there are no great differences between the Atmos weather station and the canopy level sensors. Then strangely, in November, when only plot 1 is covered, the leaf wetness duration measured by those sensors is actually lower compared with the weather station and compared with the uncovered plot. The next time frame shows the time from December to February, where both plots were covered. And here we see that it's probably not the cover that influences the leaf wetness duration in plot 1, because if the cover at the site was to lower the leaf wetness duration underneath the cover, we would expect to see the same in plot 2 that is now covered as well. But the pattern is just the same as before. And then when the covers were removed in February, we still see that plot 2 has a higher leaf wetness duration compared with the weather station, and plot 1 is about the same or a little bit lower than the weather station. So also here we don't see a clear effect of the row cover on the leaf wetness duration, but it's slightly different from what we saw for the other two sites, because plot 1 always seems to have a lower wetness duration than plot 2. We think that this might be an exposure effect of the plots. Plot 1 is exposed to the primary wind direction, so maybe because it's on a prevailing wind side, it just dried out faster than plot 2. Unfortunately, we don't have wind measurements equipment at canopy level, so this is more like a hypothesis at the moment. But this site shows for sure that there might be large differences between sensors in the same field. Relative humidity is not a factor that goes into the disease risk prediction model, but it's still interesting to look at how the average relative humidity is at canopy level or underneath the row covers. So again, this is the familiar chart with the covered period between the two blue arrows, and the average relative humidity is actually higher during the covered period. It's also mostly higher in the canopy in general when no covers are deployed, but it's more obvious during the covered period. And again, we see that this can be different from site to site. So this is our Virginia site, again the covered period between the two blue arrows, and we see that the average relative humidity was always higher, and it's not necessarily more pronounced by the covers. So at this site, the covers did not increase relative humidity. I talked a lot about temperature and leaf wetness durations, and now I would like to have a look at how this translates into infection risk days and fungicide applications. This table shows the number of fungicide applications in the 2019-2020 season for the three sites. We have the three spray schedules, the weekly calendar spray schedule and the two model-based spray schedules. With the model-based spray schedules, the number of sprays could be reduced by, on average, 63 and 49% with the Atmos and the canopy spray schedule respectively. If we look at the days with moderate or high infection risk, we see that there are more infection risk days for the canopy level sensors compared with the weather station sensor. So the canopy level sensor is more sensitive, which we also saw in the leaf wetness duration and temperature. Both factors were increased at the canopy level compared with the weather station, which directly translates into more infection risk days. However, more infection risk days don't necessarily mean more infection events. So an infection event is something calling for a spray. There might be 5 infection risk days in a row, but the first day already calls for a spray, and then the spray is good for about a week, which means that 5 days are only one infection event. And this is the same as if we only had one day that had a medium or high risk. That's also one infection event. That's why we don't see huge differences in the infection events between the weather station and the canopy level sensors. Reducing the number of fungicide applications is great, but of course we also need to look at the yield and the disease incidence. Those are the results from the variety Chandler from the Y trial from last year, and this is the average over 7 picks that we made. In the right-hand side picture, you can see that anthracnose pressure was really high. So this is a sample of berries from the control, and almost every berry has a lesion from anthracnose. We didn't see a lot of botrytis. This is why this chart is only about anthracnose incidence. The blue bars are anthracnose and the red dots are the marketable yield per plant for the whole season. We have our 4 treatments, the Atmos and canopy level spray schedules. Both received 3 sprays and those were also applied at the same days. The weekly calendar spray schedule received 9 sprays and the control wasn't sprayed at all. And we see that the control, of course, has the highest disease incidence and the lowest yield, and the differences between the calendar spray program and the 2 model-based spray programs are actually not that high, considering that we only used about 1/3 of the sprays. So although we have a slight depression in yield and a slightly higher incidence in disease, this might just offset for the fungicide that we saved, because we only used 1/3 of the sprays compared with the weekly spray. For the Virginia side, the yield results look quite different. At the Virginia side in 2020, we only used an organic fungicide. It's called Regalia and it's an extract of giant knotweed. It's a plant growth and defense promoter that is labeled for the control of botrytis. However, in our trial, we saw that the fungicide didn't control the disease at all. In fact, we had 5 sprays for the canopy-based spray schedule, 11 sprays for the weekly calendar spray, and 3 sprays for the Atmos-based spray schedule. Despite those differences in the number of sprays, the yield was not greatly influenced by those spray programs, and also the disease incidence was quite high. So all in all, we can say that the fungicide had no effect in this trial in any of the treatments. Actually, the yield is lowest in the treatment with the highest number of spray and highest in the control that didn't receive any sprays at all. This might be due to the increased moisture that came in with the fungicide sprays combined with a limited efficacy of the fungicide. So this year we will use Captan and hopefully we will see more differences between the treatments. Summing up, disease prediction models can help to reduce fungicide sprays. This has economic and ecological benefits, but most importantly, it helps with fungicide resistance management. Fewer sprays just mean less selection pressure on the pathogen population, which means that the fungicides remain effective for a longer time. And all in all, the sustainability in strawberry production can greatly be increased by using those disease prediction models. We saw that at canopy level, the conditions might differ from those recorded at the weather station, which means that it makes sense to consider microclimatic conditions for the disease prediction. And also, those sensors provide additional benefits to the grower. For example, to look at frost protection, the grower can see the temperature in the canopy in real time. It can also be used to monitor the growing degree days to see how much advanced the crop is in the growth stage. And we also saw that the floating row covers alter canopy level microclimatic conditions. We saw a higher temperature underneath the row covers for all 3 sites, which was kind of expected because that's what row covers are used for. The leaf wetness duration is also important for the disease prediction models, and we saw that this was not much affected by the row covers. But in general, if we just look at the canopy, the leaf wetness duration was increased and prolonged in the canopy in 4 out of 5 trials that we conducted so far. We also saw that there's a variability between the canopy level sensors in the same field, for example, the inner versus the edge row. So it's just interesting to see how those microclimatic conditions differ even within the same field. So, concluding, we have to say that we still need to get more insight into the factors affecting microclimatic conditions. There are just so many things like the shading between the plants, the wind direction that could have an influence that we are looking at more closely in the future. I thank you for your attention and our collaborators for conducting the trial at the 3 different sites. Dr. Hu and I will be here after the presentation to answer any questions that you might have.